So let's begin with the following. There will not be a lecture next Tuesday because of the student assembly. There will not be a lecture on Thursday because of the Ascension Day. So no lecture next week. The other aspect pertains to the videos. The videos are not online yet because Carlo has a problem with the video creation. And we don't quite know what the problem is, so he has essentially 2 lectures to do. The one for the computer sciences, everything works, and the one for you guys doesn't work. So there's something in this room that is sort of making this camera do the wrong thing, or the computer that handles the information that he processes. So the problem is he cannot export it from the program in which he renders the video, so he cannot export it. Let's see. So it may be that, so one suspicion that we have is that in fact it's related to the flash drive in there, that there's a faulty flash drive. He sees some of the video, but he cannot export it into a different format. So as long as you go to his screen, you can see the videos, but it's not very practical to get it to anybody outside of the room where it currently is. So the fault on that flash card then would not be of the kind that there's no video. It just would sort of prevent the program, the post-processing program, from doing something. Again, he uses the same for both lectures, and twice in a row. For this lecture it didn't work, and twice in a row for the other one it was all fine. So maybe that's your fate. From here on there's no camera. He's working on that. Is there anybody, by the way, in the room who has some expertise on Final Cut Pro? Oh, okay.
Why don't you use another program?
We are sampling through that. So the historical answer for that is that everything we tried had some issues. And we did not choose the program that didn't have any issues, but we chose the program that had least, or for what we wanted to do, had the least issues. And Carlo is new to this, so he has no experience in which one to go to next. Maybe involve Jonas, but this is not as trivial. I don't know whether you have done video editing. So if you go to the simplest way, like iMovie, that's the real simple one, and that is essentially straightforward. But the moment you get into more professional material, it gets more complicated. The real simple one did not initially work for doing the right transfer of the formatted, for different formatted files on different resolutions. Okay, alignments today. Next week nothing, and the week thereafter we get into the discussion of comparative modeling. No, we actually still continue with alignments first. Sciences of communication. Communication starts with asking questions. Please ask questions, keep me happy. Why do we compare 3D shapes? Because that relates to function. That's just a flashback. The details of the function have in this particular case to do with forming a dimer, but that clearly is an aspect on structure. So we also compare structures because we can, and we do it because we get further than the comparison of function, and that is therefore a reasonable first starting point. How do we compare structures? We simply compare 3D objects. And this is fairly straightforward. There are some issues to remember. It's not that trivial to have a metric in this comparison. There's a way you can enforce a metric that comes from Manfred Zippel that allows you to automatically compare big objects. In this particular case here, in fact, we look at the comparison of aggregates of proteins. It's not trivial to do that. And the major problem is partially in the way we put these comparisons together, in the way we go we move around between global and local comparisons. Structure prediction, structure itself exists on different levels. Information content 3D, 2D, 1D. 3D, 2D, essentially the same information. You completely reproduce the 3D structure, almost completely, from the 2D contact map. Although, in terms of information density, of complexity, this is 2D. This is a matrix, not 3D information, okay? You could imagine 3D objects that cannot be transferred. Most 3D objects can. Certainly the regular objects that we have in proteins can. When we go same amount of reduction, one dimension less, from 2D to 1D, you lose connectivity, you lose distance, you lose a lot, but you still have information that is important, secondary structure, solving accessibility, and that is what in 2 weeks we will talk about. Sequence comparisons, pairwise methods. So How similar are proteins? Let's just assume these were, okay, here's a letter that I do not have in my protein alphabet, so I'm not talking about proteins now. I'm talking about real-life objects. Well, proteins are real-life objects. Objects that exist on the whiteboard, not in biology. Opa, puppy. So when I align them, immediately, again, you see it's your intuition, it's your past experience. You immediately move things around and you see how they fit, and this one fits, and this is all trivial, and you can keep going. And you're all very sure about this being the right alignment. Now, some of you also will sort of attach to the string some meaning. That has a meaning, that has a meaning, and those three have different meanings. So again, I understand that in some languages things are complicated, so some language may have this as father, but several languages have this as grandfather, this as grandfather, this as grandfather. So the point that I want to make here is, on the one end where you align a string, you imply similarity, and you imply that certain pairs here are more similar to each other. And with that similarity, ultimately what we want to get at in protein sequences is meaning, is function, is structure. In this particular example, you immediately see the meaning of the word is not necessarily the one that is the most similar one in terms of the letter identity, right? So we need to do something else in it. In this particular case, you could argue, okay, these three I want to align. This one here is still related in some sense. Typically it's a historical thing, right? You're a father, at some point you become grandfather, and typically you don't become grandfather without being a father. So there's some story to this. This is something else that you don't want to align in there. But ultimately what is right in the alignment is defined by your expectation. That expectation is written in what is called the objective function. What is an objective function? This is essentially what you call your criterion to optimize. In this particular case here, the criterion for optimize that I put in your heads with these slides is simply a match of identical letters. You sum over all the letters that match. Let's hold it right here. Bioinformatics, computational biology, is a lot about the many freedom, points of freedom. So we have, in some sense, you can imagine this like two extreme worlds. You have the worlds of high energy physics, where you essentially, or gas, where you describe the gas as something that is described by infinity. You actually, in every gas, have a limited number of particles, but the number is so big that your idea of infinity gives you the right model for the gas, right? On the other extreme, you have your physician. Your physician sees you, and you are the case. Maybe there's some experience, but there is essentially an experience value with respect to the disease that you come with of 1. Or when you look at computational biology in medicine, it's not 1 but 10 or 15. They have very few test cases, and they try to generalize from very few test cases. So that's one extreme. And biology is very often on that side. We have a few measurements. Doing one experiment takes a long time. And physics is very large numbers. Most of what we guys do is in between. As long as we have 5 measurements, the experimentalist will handle the data. You come in only because the experimentalist no longer can handle the data. It's not infinity. It's not a lot on the scale of physics. But it's just too much to look at. It's something, in biology we need the individual example. But the data that you're presented with is too much for the individual example. Yet, you have to boil it down to explain it to the one who cares about the individual example. That's the challenge. In some sense, on the level of scale of what you are challenged to do, is a world in between two extremes. And there's no other discipline, I would argue, that has the same challenge. Okay, now, part of the problem comes now in the freedom of certain parameters to choose. It all begins with something that is totally trivial always. And that here, in this particular example, the objective function, you just count the number of identical residues. Is that a clear definition of the problem?
Not really, if we want to get some information about the function.
That's not what I mean. I'm sorry. I'm on a much, much simpler level now. You're right in your answer. You're 100% right. But I meant something much simpler than that. So I say to you, write the algorithm that compiles the number of identical letters on the whiteboard. Okay? And give me the number. Now, can you imagine that, in fact, there are different answers coming from this group? So presuming that you're all not sleeping, you all look very, very awake, you're all there, you're intelligent, you all understand what I say. So that is not the problem. But could you imagine that you can take what I say and interpret it slightly differently and come with different numbers? How? What can you think about? It's on the whiteboard.
Do you mean to hold the sequence or to split up the sequence in parts?
I have those three sequences here, and I want you to tell me what's the number of identical letters.
That are in this exact
In that example.
composition or?
On that example, what's on the whiteboard? Is there only one answer to that?
No, you can count differently. For example,
Yes.
it should be in all three, or if you have a point, then do you still count or not?
Exactly. So where do you start counting, right? Do you begin to count 1, 2, and that's it, or 3? Do you count the P because it's all three of them? How do you count this? Are you going to give me a number, or are you going to give me a percentage? Is the percentage maximum, or is the percentage, if you give me a percentage, is that a percentage of where the dots are? It's a totally different problem, a simple problem here. And it's absolutely, what I say is absolutely clear for every one of you. And you will all conclude that it's trivial, okay. But actually, in this trivial detail, there may be a difference. It may work differently. Your alignment, yes, you're right, you want to get the function. Maybe you get the function better by beginning to count where all three begin. You have to test that. And the problem, again, the challenge in computational biology is that usually the number of free parameters that you make are beyond of what you can test. Or another typical challenge is that the moment you write the code, you will realize that you now have a choice for the two parameters, but you're writing the code because you want to collect the data. So you don't have the data yet. And by the time you get to the data, you have some kind of other problems. You forget about that choice again. But you typically have to make a choice that, in fact, very often is not really the right or the best. And you're very often in a situation where you have to have the best, quick, fast guess. Computational biology is a lot about hacks and about having an intuition for the right guess, thinking it through what is a good guess. Anyway, that is part of what I wanted to say here. Let's get back to the objective function. And the simplest objective function, for instance, again, now I ignore again the problem, and I assume that saying percentage of letters that are identical is a clear definition. We established now this is not the case, but now I pretend it is. And I had given you all the other words that I need to make it a formula that is clearly defined. But then again, what you said already, functions may be described by more complicated ways of looking at it. Well, macroscopic similarity, ultimately what we are after is, you say, function, is structure, and is in fact evolutionary relation. Now, for the time being, let me just continue on the issue that is pre this one here. On the issue where we don't have biology, where we still compare only letters, the string matching. In the string matching, so we have these two strings, we want to match them, and all we want to do is, given some definition of optimal, find that optimal solution. Actually, again, it's the same problem that we had before. Even at that level of simplicity, we immediately run into the next issue, which is the issue between global and local. So do I, in fact, align from the beginning to the end? Or do I align just fragments? The moment I say I align fragments, I get into the new problem of how to cut. I will get into that. Let's ignore that. Let's get back to what is an insertion. If I aligned it end to end, or in this particular case, I would clearly have to insert. What's the interpretation of an insertion? You all have seen this dot. And you have to always remember, I mean, this is in some sense symmetric. So the dot here is as if I cut the A here, provided the alignment is right? But what is the interpretation of in biology, sequences don't have insertions. So if I do an alignment, I imply similarity in function, I imply similarity in structure or evolution. What do I mean by a dot?
Over time it changed?
Yes, but what does it mean? There's a dot. So over time, there was suddenly a residue invented. And how do I picture that in a structure? How do you believe? How do you picture an insertion in a structure? What do you believe will happen? Or do you have anything in your head, in your mind, that would sort of visualize a dot? Yes, a new residue is inserted. In this particular case, if we assumed that OPA is the first historically, then the alanine, the A, is inserted. It's made a little bit longer. This little bit longer, what will that lead to in a structure? I say it would be like 3 or 4 residues. Now the common interpretation, in fact, is that we have sort of a loopy thing, right? So something. Remember when I looked at the comparison of structures? There's a core, the core is similar, and then there's an embellishment, as Christina Rengo calls it. It's something that comes on top that may be related to function. That in fact may be exactly the binding site. Antibody is an example for that, where you have a long loop that is so flexible and can do so many things because exactly it's the long loop in its local environment. But the interpretation here is simply you do something on top. In fact, the immediate interpretation here would be you have something that is added here. The historical interpretation is, however, the evolutionary relation that implies similarity in structure and function. And I made a mistake in the way I organized it. Now I'm back to the biology. I said I would only compare strings. Well, I swap. My talk is not optimal. Getting back to the word homology. What does homology mean?
Some kind of relationship.
Hey, that's true. That's good. Yeah, that's a great start. Anything more? Anything that goes a little bit further than some kind of relationship?
Ancestry.
That implies similarity.
Similarity in what? He says ancestry. So is that what you mean? So similarity. So we have two. I have 2 words in the room. Similarity, and that immediately in your head does something like similar in structure, similar in function, similar in something, right? Well, ancestry is not necessarily the same thing. Is it?
Sometimes not.
Well, in terms of, yes, another thing. So what you do, let me just be very provocative here. There's a scientific procedure and there's a journalistic procedure, or the survive in life. The best way to survive, you cannot be a good scientist without surviving in life. There was a generation, I believe, when you could be isolated in some island and do your thing. I believe largely that is over today, certainly in computational biology. So first of all, you have to work on your intuition and on your normal. You have to be a human being who can stand on their own feet and live and do it and be right. Okay, that's very, very important. And for that, you have to have guesses, just like you did, that have to do with probability. It's a relation. It is sometimes, sometimes not. The obvious things, say them. That's always important. But scientific is you try to look at the information content. In the sense, for the statement, sometimes yes, sometimes not, since that applies every time, against background, the information content is 0. So actually, I'm just provocative, right? You have to learn both. So, historically, the word really is philo, race, tribe, kind, genesis, birth, origin, and we get to, can anybody, well, okay, it's a very, very bad copy here. Do you see anything more than some little bit of white that has something in it? No, you can't. Fair enough. Now I unfortunately have the quote. So you see a little bit more here. That is the tree of life. as it was pictured in a book from 1859, The Origin of Species, and that, in fact, in that very book, is the only figure in Darwin's book. Has anybody read it?
I tried.
Yeah, that's as much as I read. Maybe I did a little bit longer than you, but it is, in fact, and I believe ultimately for us today, part of the problem lies in the fact that he tries to beat what we would call a dead horse. So most of his message we accept so much today that we find it boring that he has to explain why that is true what he says. That is part of it, boring. But part of it, I believe, is just because it's lacking. He describes images. He describes his animals and the similarity between animals just in words, and it gets complicated, and that means it gets boring, and that means I tried, so I cannot continue. Anyway, so I like to present Darwin in a context where I say something changed and something is the same. That clearly has changed. By the way, on that note, what is always the same is this was published in '59. Darwin was not the first. It was pushed to be published on one day, in July 1st, I believe it was, along with the other one, because the other one was there first. Both authors of both pieces were not present at the point where it was read, the paper that established it, The Origin of Species. It was read by some secretary who felt that Darwin's work should be pushed to sort of be competitive. And that kind of thing is still the case today. Anyway, let's get back to the tree of life. Bacteria, archaea, both of those are prokaryotes, eukaryotes. So the eukaryotes have the nucleus, as you know. Here's just a different way of looking at the tree. In this different way, we have this one applied here. And now looking at the detail of these two, you see that, in fact, what looks upon first sight very, very similar, has some different interpretation as in to where the root starts. The root here is they all have one common ancestor, while in this particular tree the prokaryotes come together and then have another. Prokaryotes and eukaryotes are one common thing. Let's get back to the speciation event that somehow starts it all. One way of looking at it is you have these two flies that swirl around, and more flies, and some of these have offsprings that create a new species. It's typically not that simple. Typically you have a bunch of flies that create another bunch of flies, and some of these are a new species. And then you build a species tree and the speciation event. What's the definition of a species in biology?
They can reproduce.
That is very close to it, not 100% right. So the definition is not the offspring will in fact be fertile. That's the definition. So in some sense what you reproduce means having offspring. It depends on how you define it, right? Reproduce could mean I have offspring, and offspring you could have between species, but the offspring typically is not fertile. So the offspring will not have offspring. Okay? In the sense of longer section of evolution that implies one can reproduce and one not, but okay, that's the biology definition of it. And homology, in fact, is defined on that level. It comes from homo logos, agreeing, logos is the word, and it goes back to Richard Owen, 43. It's the same organ in different animals. It's like, and this is sort of the diagram in 47 that shows the conceptual archetype of vertebrates. Here we are. Homology is defined on the level of organs. Okay? So we have organs that are similar to each other. That is the word homology. That is the way it was introduced in the language, and that is sort of the original meaning of this word in the context of biology. Then it got into genes and implied, in fact, that they originate from a common ancestor. That's the definition of homology. Now, there's two different ones, orthologs and paralogs. Orthologs, they come before the speciation event. At the speciation event, and paralogs are duplication events afterwards. The implication of that is orthologs is essentially, in human and mouse, the corresponding protein that does the same thing. So orthologs then are things that are related and have similar function, similar structure. And paralogs are things that duplicated. Since I have two versions, I can evolve a new function. So they may have different functions. That is the basic idea behind it. And reality looks very different. Now again, let's get back into the speciation event, how I picture this. I like those slides here. So you have these flies being happy and doing their thing and mating and munching and doing whatever they do to be happy. And then, disaster strikes. There's a river or there's some water coming, and the water actually separates out them poor flies on one side and the other side. This is tough for them. But good thing is they still have enough here, and on one side of the river you have a little bit different food sources than on the other side of the river. They begin to have different preferences for food. Different preferences for food, you may sort of have realized that, lead to different preferences for mating. So it's much more fun when you love somebody to also eat with them, not when they have totally different habits in eating. And one day you come with this banana, you're happily munching on your banana, and you swim over, and you can no longer mate because you just have different preferences. In some sense, that is the model for evolution. That's the way speciation events happen. And you can actually do that really in a cellar. That's why Drosophila is such a great model organism, because you can have Drosophila in your own apartment. No matter, even in the New York apartment, people had Drosophila. Flies that they use for doing experiments like this to reproduce exactly this evolutionary behavior. You can actually do that in a little cubic meter of your space. So that's just the analog of a few books. So again, biology defines a species as no offspring without passing on the genetic information. Then this is biology, so there are always exceptions. So there are hybrids that have children, there are same species different species that interbreed, and there are all kinds of issues in between. One issue that you all may know, everybody in this room here has 2 to 4%, 2% most likely, everybody 2 to a little bit more percent Neanderthal in them. If we would have an African in the room, they would not have that. We all agree that Homo sapiens and Homo neanderthalensis are different species, so they wouldn't have offspring. If the definition would be strictly true, we wouldn't have Neanderthal genes in us, right? We do. So that's the limit of biology. So obviously there was offspring. In fact, now they know, and that's the group of Pantes Febo. I really go Svante Pebo in Leipzig and Janet Kelso, and from their recent work we now know that in fact most likely there were two events, and most likely they happened about 50,000 years ago, two mating events. Okay, back to Richard Owen, back to the issue of homology that is defined on common ancestry, is misused to identify similarity in protein function and structure. Many sources you will find the word homology being used to say similar in structure, similar in function. This is not the original meaning, okay? It's a misuse. We should rather say similarity in structure and function. Anyway, this is very frequent, but one aspect of this mistake is it keeps reminding us of the evolutionary relation, and that is an important one, and we get back to that. Again, comparison of sequences in order to find evolutionary relationships on the level of proteins. Okay, what we also want is similarity of structure and similarity of function. That ultimately is what we are after today. Again, somebody in the room had this question, why do we compare structures if we want a similarity in function? Because structure implies function. Dynamic programming is essentially brute force, and that is what we did on the whiteboard. Essentially what you do visually is you move these sequences around until you have a top hit, an optimal hit defined by the objective function, and this is exactly what this algorithm from Needleman and Wunsch published in 1970 here in Journal of Molecular Biology achieves. It guarantees that two sequences are aligned in an optimal way, provided you don't have gaps and provided it's a global alignment. The way you do it, you simply move around this string, put that into a matrix, compile all possible points. In this matrix, you can only walk on the diagonal. In this particular walk, that is the best hit, and you cannot get better. If you want to get better, you have to, in fact, allow gaps, the insertions. With the gaps, you can go further. You can do alignments beyond that. You can, in this particular example that I have, you can reach a score of 4 optimally. Typically, when you have longer proteins, the profiles of the alignment profiles, the dynamic programming profiles, look like this. What I mean is, you have regions here where you have very clear one solution examples, and regions where alternative solutions are totally possible. So the more divergent two sequences are, the more unclear the alignment becomes. It's still, there is one optimal solution. Okay? But the second to the best may be as good. And the difference between the best and the second to the best gives you an idea how stable that best solution is. If all of that would, in this particular case here, be close to the diagonal, you would argue, okay, there are different solutions, but you don't really care. If, however, one solution would sit here and one solution would sit somewhere completely different, you would argue, well, there's something that I don't know about this alignment. There's something that could go wrong. Sometimes you could use such a matrix dot plot here to have an idea about domains. But now we get into the nitty-gritty detail of alignments. That's beyond what I wanted to do. Much simpler. In terms of the objective function, typically, you would, for instance, measure the identity of residues and would possibly introduce a residue called dot, or put a penalty for having a dot in there for an insertion. In this image, these two would be equal. Whether you have 5 dots in a row, or you have one dot here as dot, residue, and so forth. 5 dots. It's the same thing. If gap gives you a penalty, they both give the equal penalty. Are they equally likely in biology?
No.
Why not?
Because insertion and deletion events are more likely to happen consecutive.
Why?
Because there could be large parts duplicated.
And we are back to the interpretation. So that is exactly what I meant. So typically the image of an insertion is a loop. So you don't have a one residue loop. It's much more likely to have a 5 residue loop, where in the previous one you had nothing, then to have a little bit of a dent, a little bit of a dent, a little bit of a dent. Because this 5 times dent in a row would completely blow up your helix strand or whatever structure you have. This is why this is very unlikely. Yes, indeed, in alignments, when you look at blocks, you see this block alignment idea, and that in fact is where Stephen and Jorge Hennickhoff got their idea of blocks for. So you have regions where you see something is aligned, regions where you see, well, these two proteins are aligned, but most of those don't, and then really regions of higher density. If you plot the density of the alignments, just the number of count per position, you see that there is a clear function, and there are regions that are more densely aligned, have a higher density than others. How can you put that into this equation here, the Needleman-Wunsch equation, that simply sums over the number of matches and gives a penalty? How can I change this formula in the simplest way to force the biological observation into the formula?
Increase gap penalty?
You make this number higher. Will that do it?
No.
All it will do, it will reduce the probability that you open a gap. But the probability of residue gap versus residue gap residue is still identical. It is still the same subtraction for the same number of gaps. Any other idea?
You could reduce the penalty for following gaps, maybe?
That's what people call a gap opening and a gap extension penalty. So the following gaps is essentially the extension. So you reduce the gap extension penalty. this is sort of a rule of thumb kind of thing. Gap open is 10 times gap extension. And with that, you simply add another term here. That is simply the extension penalty. And you have this formula, and that formula immediately, again, you simply wobble a little bit with the objective function, and you're still in dynamic programming guaranteed to get the right solution. And this time, you even enforce a solution that is biologically more reasonable. All you did is a minor change here. That's remarkable, right? Now, we have this dangling issue here of local versus global. What is best? And that brings us to the question that we had before, and we now leave the strings in some sense. Is really the total number of matches the best, or is it something else? What should be the units? One aspect of the unit here, since we have 60 to 400, proteins are from 60 to 400 residues long. Single domains typically are from 60 to 400 residues long. You immediately see that the answer most likely will not be a global alignment is best, because it will be very difficult to map subfragments that are related, to find the commonality in things that are so different in terms of the length. Let's just, I mean, let's imagine we would be a species where some of us are 60 centimeters high and some are 4 meters high. Just think about that every day. And the difficulty of walking into spaces, of sharing the spaces, this is a remarkable difference, right? Anyway, ultimately we will get into a situation where we have a question such as this. Here we have the GQ mapping to the GQ, so we have 2 and 2 right. Here, in this particular case, we can extend further the alignment, and in that further extension we would get to a point where we have 4 and 7 right. That's 100%. That is just barely a little bit more than 50%. What is better? But this is 4, and this is only 2 matches. What is the better solution? And how could I possibly get to the answer to that? Any idea?
You can look at the fact how much sequence identity is needed for this similar function, for the same function. And let's say if you have 50 or 60%, then it's good.
That's a great idea, and in fact I'm going to show you a couple of slides that answer my own question in a way that is completely compatible with what you say. But since my role is the role of a teacher also, I'm just going to take what you say and turn it around and sort of punch a hole into it. That's again a scientific procedure, right? This is 100%. Okay? No matter what you find, you will never get beyond 100%. I mean, 100% is sort of typically the end of your story, right? So then it will always be the best. Now, say the best story for a matching function structure would be 70%. Okay? Then you would argue, well, 2 and 2 is higher than 70, 4 and 7 is not. And assume that everything so far, I did what you said. I established it has to be 70%, in my head at least. And would you then believe that answer? Okay, the 70% is not right, it's going to be a different number, but whatever the number is, I can carve these examples such that the story that I'm telling you in principle is true. What I'm talking about is the 2. 2 and 2. That's tiny, right? So do you really believe that we will ever, if we have 2 matches of just a fragment of 2? Yeah, no matter what percentage sequence identity is, 2 is always too little, right? This is most likely, I believe we all in the room are happy with sharing this view. That 2 is such a small number. We have 50 million sequences. Any pair of amino acids will be in the database. Most likely in different contexts. So this will never be enough, right? So in some sense what you propose will give you an answer that in fact will not quite answer that problem. But we're getting close to it. Here's another issue, and that is the issue of what is the best criterion for optimal. And that brings us to the substitution matrix. And the substitution matrix, again, when you run a typical blast here, blast output one sequence, one other sequence, the identity is shown here in between. Here are in this particular case, there's a potassium channel. The gating, the part that is important for that potassium channel, is in these three residues. All three residues are not identical. Whatever is in yellow here is not identical. And that immediately, well, actually this one is. That immediately gives you some idea that maybe there's something else that we could look at, and that we in fact look at a scoring matrix. Any idea what the idea of a scoring matrix is? A substitution matrix? How could we refine from, now think about biology, from the letter matching, from the simple word letter matching case?
The physiochemical properties.
Yes, that's exactly what we see here. So we see that we have, in fact, lysine and isoleucine, we have two hydrophobic residues. And it's fine that they are not the same letter because they essentially have a similar biophysical feature. That is acceptable. How would I sort of formalize that? Well, one way is I could just say a certain set of residues are hydrophobic, a certain set of residues are charged, and put that into some ad hoc table. Any other way in which I could do it?
Have a matrix with how interchangeable each pair of residues are.
But how do you get that matrix? That's my question. Yes, that is exactly what we are after, but how do we get it?
From a list of sequences, for example.
How do you get the list of sequences?
I suppose if you want to have a general, you would a general matrix that works for all sequences you provide, and as many as you, a large data set with very diverse sequences. And
But what do you need? So you take a set of sequences that have nothing to do with each other? Okay, anybody who don't know, I mean, she was the first to answer.
Take the same protein, enzyme protein from different sources and then compare, and you know this function is the same, and compare the sequence, and then you can see what changed and what is possible to change without.
Yeah, exactly. So you need a set of proteins that you somehow know something about that allows you to state that they are similar. By, for instance, the enzymatic function. They do the same thing in two different organisms. The first, does anybody of you happen to know who was the first who came up with this idea? Okay, you don't know the name.
Should.
We all should. I don't care about should. You shouldn't. You absolutely should not care about should. You have to learn to think. You're not memory storage machines. We have a computer for that. Thinking is what you should learn. And take your time to think. That's what you really should do. Everything else is nonsense. But now back to the question, any idea about it? You don't know the name, I don't care. What about the time?
60 years ago?
60 years ago. Oh, that's a long time ago. You're getting not that far away, so we're getting close, at least from your perspective. I take it for you, there's 20, that's sort of your age, and a little bit more, and then there's parents and grandparents, and this is all longer ago. So grandparents is like, and this is like that, right? It's a long time, 60, 80, 100 is no difference for you. 78. The person was Margaret Dayhoff. In 78, she built a library with all the then known protein sequences. That is essentially 71 families. And overall, in this entire set of these families, they were very, very similar. Because overall, there were only 1,500 mutations, variants in there. Essentially, a bunch of very related proteins. Okay? That's the atlas of protein sequence and structure, as she called it. Let's get back to history. And here we get back exactly to your 60-year number. Fred Sanger does the first sequence on insulin. The first structure is 58 and 59, Kendrew and Perutz. And then, actually, it takes another 20 years before you have that set. Okay? This is the time it took. And Margaret Dayhoff was really the first person who had the guts to sit down and completely try to classify this. And the classification was not so much really driven by the one-to-line sequences. That evolved at the same time then as a means to compare sequences. But her thing was really get an archive of what we have and understand evolution. That in fact is exactly why the matrix she evolved. Now she has this bunch of related sequences. Now she can simply, as you said, count. Or how often do you have an A go to a G? The PAM matrix, 1 point mutation per 100 comparisons. That is history. Wikipedia, all kinds of sources on the web talk about PAM because it's very intuitive. It's the beginning of it. It's important historically. Has absolutely no reality today. If you see anybody using that, turn the other way and do your own thing. This is, since before you were born, is outdated. Okay, your parents didn't use that if they were in the right mind. Today, there are tons of people run around who really believe this is. Anyway, here's another way that is old too, but that is the modern way that is completely still used. Blossom, blocks. Blocks again comes from this idea that what you see is regions that are entirely aligned. And comes from here, what I show is Stephen Henikoff. Jorge, his wife, is the mathematician. He's the, as you can already see, the pipetter. He's the experimental biologist. And again, it's the same idea. You take a bunch of proteins for which you know that they have something in common, and then you simply have a simple mathematical way of counting the log odds, of counting what is substituted at what position. So you have the 20 amino acids here, the 20 amino acids here, and then you essentially do log odds. So you count, translate count to information, put the information here, has a negative and a positive. Negative means less than expected, positive means more than expected. In this particular matrix, you see that in matching an E on an E is 4, that matching an A on an A is 2. So the diagonal does not always have the same value. The tryptophan here, you may note, has a much, much higher value. Why would that be the case?
It was very seldom observed that they eject.
Yes. So then it's true, right? It's very, very important to match tryptophans. Here's a reason why this could be not quite true. It's true. We very rarely observe tryptophans to be mutated. Do you know anything on top of that? Tryptophan is a rare residue. So it is true that you observe tryptophan less often than all the others. So some of that rare observation of tryptophan not being a tryptophan has to do with the fact that you don't observe the tryptophan very often. And that is in fact highly non-trivial to normalize out of that matrix. And this matrix is not contained. And I'm not even sure whether anybody has tested whether if I normalize that effect out, I would do better. Possibly not. I should. So here's another nice story, and I should have just put up. This paper here, Blossom. Again, this is over 20 years old, but this is still in everyday blast comparisons what is mostly used, Blossom. Blossom was published in '92. About 20 years later, somebody published another paper showing that in the original publication of Blossom, that has been quoted I don't know how many thousand times, there was a mistake. There was actually a mathematical mistake in the formula, and that formula led to the wrong programming. And that led to a mistake in that Blossom matrix. 20 years later, somebody spotted that. Not only that, that person actually then had, of course, the clever idea of fixing the mistake and trying out how well the fixed solution works. Lo and behold, you guess where we are going with this, right? The solution with a mistake performed better than the solution without a mistake. True. True story. I should pull out the publication. It's one of those remarkable stories. Today, there are many, many, many more substitution matrices that exist, but still Blossom 62 is what is mostly used. 62 Anybody has an idea what that comes from? Essentially, it has to do with the fact that the set you initially aligns is similar to each other at the level of sequence identity. Everything there has more than 62% sequence identity. Okay, so when you, in fact, want to align proteins that are more distantly related, you tend to go for a Blossom 35 or whatever is the next number. So there's a bunch of Blossom matrices done. I can't remember what the next one is. If you wanted to use a nice graphical interface that allows you to play around with dynamic programming and changing gap penalties and things like that, there's a nice interface, a nice program done by Francisco Melo, Santiago, available here. Dynamic programming is optimal, but there is one issue that has to do with time used. Dynamic programming is taking time that has to do with the square of the length. Okay, square of the length. We have proteins that are 400 residues long. Is that a lot? If I want to align two, say, 400 residue proteins on a modern computer, is this a major issue? Who is with him?
What did you say?
Huh?
What did you say? No.
No. So it's not a major issue. So today, dynamic programming has no more limitation. But what you forget is we don't quite do two proteins. What we actually do, and this is an example of a story that essentially we came to Germany in 2009. In 2009, in September 2009, we had about 8 million proteins. 2014, when I did this slide, we had 55. We still have 55, that's a different story. This is 6.9 per month, or double every 2 years. The double every 2 years for computer is gone since 90 something. Okay, this is faster. Okay, that's problem number one. But actually that is not the real thing we have to do. We do comparisons. The comparisons means that actually I do an all against all. And in all against all I have 64 billion here in 2009. Now I have over 20-fold increase every 2 years. Because actually what I need to do in order to do an alignment is not this, it's this. Okay, this one was already faster. Oh, yes, it's 10 times faster. So I just wait a little bit longer and everything gets more complicated in the world of biology. And that really is true. This is a truth that began to hit the large centers like EBI at Hingston or the NCBI about 2008 roundabout. And today, nobody really can handle the data anymore. So it's a major issue. The way we all handle this is we reduce by throwing things away. And this is a hack that we have to live with, but this creates increasingly problems. By the way, there also is this issue that pertains to the storage space is not as dramatic as this one, but still in 2009 we came here with 5 terabyte, and in 2009 5 terabyte was still easier. Not everybody had 5 terabyte, so even in this building very few people had that. But still 5 terabyte in 2009 was easier to handle than the 300. We have over 300 terabyte now, and that you write it to a disk at LRZ. We tried the other day to have a backup from the tapes. Any idea what that takes? You have a tape backup of 30 terabyte. 10 terabyte took us 2 months. That's, we have 300. We gave up and we just reproduced the data. But we really get into issues. Computers no longer handle these amounts of data. By that way, we sequence genomes. The sequencers that you get are as big as a printer on your desk. You can do a human sequence overnight. You produce terabytes of data every night, every day. Okay? Now there's the Broad Center, one of the largest sequencing centers. And then people in Leipzig here, they sequence, for instance. How do you believe they ship they get the data from one to the other for sequencing? Fast internet connection, no? No. This is modern days. FedEx is the way. They ship FedEx disks The large sequencing centers constantly ship disks around. So when I was your age, they had physical probes that they shipped around, biomaterial, and that was, my wife still has this issue to get with biomaterial from the US to here. It's horrible. But now it's FedEx is clearly solved, and you just ship disks in the mass. It's incredible, right? This is not, when I was your age, was not the way I imagined 2015 to be. And that ultimately is because this is a nightmare, because the biodata grows so incredibly fast. But back to the point. The square of length is not an issue when I compare a pair of sequences. You're both totally right. But since I do this on a large database, this is an issue. Another issue of dynamic programming has to do with how to choose parameters, such as gap elongation and extension, and that again, on the level of complexity it has, becomes a problem. How can I speed up? So you all, let me follow on what you said before. You all should have heard the word BLAST before. BLAST essentially is hashing, fast and dirty. The way it does it is it begins with seeds. Those are words. These words are translated into a hash, into a number, and I do a fast hashing of these numbers. Very, very fast. The first issue is you may, in fact, for instance, a typical word size in BLAST is 3, and that is about the time that we have to change that because the database now is so big. So far 3 was right. It begins to be too small. But, so one issue is if you don't find any word similarity between two proteins. Could that be? Why is that not going to happen? Why is that not a problem?
Random chance.
Yes, but you don't want random proteins to be aligned. You're right about that. So if you do the next, you're totally right. But if you did the next step, then what you're saying, actually, what you're implying by that is, oh, that's not a problem, because I will always find random superpositions and will always be able to align them. That's not what I want, right? That may be true. It is actually true. You're totally right. And that's the reason why we no longer can use 3. But this is not the comforting step here. The comforting step is the other way around. When we get to that level of divergence that we do not find any word of 3 between two proteins, then we have diverged so much that we need a different technology. BLAST, in a pairwise comparison as this one shown here, is applied for cases where we are so similar that most of it essentially is somehow similar. Okay? So finding a letter of 3 is highly likely. Okay? Because they're sort of related, not because the database is so big that you always find something. That's the point here. And then we get to the next step where we essentially expand the local seed. How can we do the expansion? The simplest way, of course, is you go one by one. You jump. You simply ask, is the next one still higher than some average? Or are you still improving over the alignment score or not? That's the simplest way. In fact, historically at some point that was done that way. Today it's different. What is the better way of doing it?
Combine nearby seeds.
Yes, that is a clever way. In particular, in the example that I show here, is immediately suggested. But let's go into this example here. It's in fact not the solution that is done, but you're right. That would be the next step of going from a simple seed, more than just going one at a time. It's a very clever idea. I would do that too. But this is not what I was looking for. So how do you align two proteins again? What's the safest way? Forget about CPU, dynamic programming, right? That's what you do here. Now, why is this suddenly helping you? So why can you, I'm trying to speed things up, and I'm going back to dynamic programming. Why can I do that?
Because the fragments sequence are very short.
Exactly. We have a problem that is in the square of the length of both. And since I have short fragments, the square of the length is a small number. even with the first split, I already have a quarter of the time. But in this particular example that I have much more than split into into two. But even if I split only in 2 fragments, even if I'm too lazy to count, maybe 60 here, the number of residues. So even in the 2 split where my word size would not be 3 but 30, even in that one I would already reduce by a factor of 4 in terms of speed. Just, remember the effect of squaring things. What is implied in between? What if I cannot, by dynamic programming, by some criteria that I put, expand in the dynamic programming? So the dynamic programming criterion that I have, or the threshold I put, says no more alignment. What is BLAST stating about the region in between? Exactly. It's exactly stating what you all say. No clue. There is no statement. So the regions in between remain undecided. Biologically, it doesn't make sense. Unless you believe that we have, in fact, something that is a matching unit, which could be a binding site, which could be a domain. But in the example that I showed you, you immediately see, well, that you have such short sequence motifs. Could be the case, but there's something strange in it. In an extreme example like that, most likely you will not find. You will most likely not find areas where you have in the ballpark of 10, I am slow, 3 times 10 that are aligned and are so similar as I constructed this alignment, and have nothing in between. So that gives it immediately away as a toy example. Or if you saw that, there's immediately something wrong. But still, BLAST may not be able to find the solution in between. In this particular example, there is in fact no clear alignment in between. So the whole example is made up such that the story shakes out. Let's get back into the people, into the players. There are historically 2 major parts of hashing. One is Bill Pearson here in Fast A, and one is the BLAST, Basic Local Alignment Search Tool, that comes from Steve Altshull. Majorly involved David Lippman, who is now the NCBI, has been the NCBI director since many years. Stephen is at the NCBI. Eugene Myers, grep in Unix, you all know. That's Eugene, for instance, Eugene Myers. The assembly of the human genome is Eugene Myers. Eugene Myers is now actually in Dresden and does and builds microscopes. So he moved a little bit away because he's trying to make images of the fly and essentially have one image of all the neurons firing, and that got him into building microscope. Gene is just somebody who has little problems and finds a solution. grep was one of those early ones. He contributed to BLAST. Webb Miller was one of the people who sort of got the world into sequencing extinct species, such as mammoth and other species like that. So it began to become really more experimental. Lippmann, Altshull, Tom Madden, Alejandro Schäfer, those people remain truthful to computational biology, and this, in some sense here, David Lippmann is still the boss, the others are still building the tools, so that still builds what we see today as BLAST, the original one for the pairwise in 1990. Fast matching of single words. The major challenge when you have these single words goes exactly back to what you said. There's a certain random probability that you match any word of 3 in things that are not related. How do you measure the distance between what you have and what is the background? You plot a score where you have the background, the hit, the degree to which your hit is informative has to do with the variance, the average here of the background, the variance, and the distance between the average with respect to that variance and the hit. That's the expectation value that you can then turn into a p-value. Now, how do I get this? I run my particular BLAST hit. What I get is this. It's an alignment score, right? Where do I get the blue from? If I want to establish how much that differs from the background, I have to get my fingers on the background. How do I get the background? Okay, let's just throw at you the simplest solution. This is the hit between two sequences. I can take the same sequence and align it against everything. Would that work? Yes, it would, but every pair comparison, instead of comparing one pair, I would always have to have 55 million comparisons. And that would make something very slow of a speed-up version of a method. That would by far lose the time that I gained in the length square. So, no, that cannot be the solution. There could be, but it's not a speeding up solution. It's a solution that allows you to use more CPU time, so that people can build new computers and buy new computers. There's all kinds of good reasons, but
can we calculate how likely is a random hit?
How do you do that? Yes?
I don't know.
You're on the right, absolutely on the right track. Question is how? Well, one way in which you could do it is you could compile it once and for all for a randomized background. So you take the database, you chop it up and you turn it, you take it into pieces, reassemble the pieces, and then do an all against all. Okay, that means that in fact now you have a lot to do once. But for all the future comparisons, provided you do a lot of comparisons, at some point your balance will be positive, because you have to really do it only once. Another way in which you could do it is you could estimate how this looks given a certain distribution of amino acids. I have 20 amino acids. You have a certain distribution at which they occur. And from that you can simply compile certain assumptions made, a probability of that background distribution. So in that probability of that background distribution, in that situation, then the probability here would always change according to the database you'd align against. And you would simply, all you need is the amino acid composition in the database, and you can essentially estimate some part of this. What you cannot estimate is the variance, what you cannot estimate is the height, and that ultimately means you cannot quite estimate the e-value. For that you also need the size of the database. Because whether you match 3 particular letters in 1,000 or 1 billion is a very different probability. And that in fact means that now the implication of what I'm just saying is that you run a blast against PDB, 100,000 sequences, and you run the same blast against 55 million sequences, and you get the same pair of proteins. Yes, that is the negative aspect of closing doors. So there's two comparisons. Query against PDB, query against UniProt. Query finds the same template, T. Is the e-value of these two findings the same? In BLAST? No. Absolutely not, because that is precisely the variance. That's precisely the probability, whether you, and it's the same hit. It's the same pairwise alignment. One appears to be much more important than the other one. And that only has to do with the background probability, and that only has to do with the way you sample the background probability. Okay? And ultimately that, in fact, is the major difference between the original FAST-A way of doing it, search-sec. That, by the way, initially was done with a precompiled distribution, also involving David Lippman. Bill Pearson I mentioned before, but David Lippman was in that one too. FAST-A does the statistics better than BLAST, the original one, at the expense of not gaining that much time. So this was just barely faster than dynamic programming. This was 10 times faster. This did poor statistics. This did good statistics. Now, for which one the value is higher? And ultimately, the answer to that question has to do how it is applied. 20 years ago, today I will get to other ways of doing it, but 20 years ago, what people did is this BLAST was so fast that you could easily run BLAST, provide 1,000 hits where you expected 5, and then do a dynamic programming on the detail of whatever remained. So BLAST gained so much in space, in time, that you could easily combine it with dynamic programming and have the best of both worlds. FAST-A, at the end of the day, took as much time, but somehow did not have any of the two extremes. And that way, ultimately, although in many ways it did the detail of the solution better, was the tool that did not survive, was the tool that was less useful. That it was, by all means, better than this, but less useful, just because there was a difference between a factor of 2 in speed and a factor of 10 in speed. Ultimately, that's what it boiled down to. And it was not necessarily clear from the start that this would be the way it would evolve. We could say that only 5 or 10 years later. How accurate are pairwise alignments? And I believe I have, at this very point, I have two options. I could just zip through it in 5 minutes, through the next 40 slides, or I could be a little bit nicer to you and say that's it for today, and that's what I'm going to do. Thank you. Any questions?